This lecture is relevant to fatty acid synthesis. We already looked at beta oxidation, which is relevant to fatty acid oxidation. Now we're going to look into fatty acid synthesis, which takes place in the cytoplasma, while beta oxidation takes place in the mitochondria. For the fatty acid synthesis, for instance, in the making of palmitic acid, it is starts off with this acetyl-CoA carboxylase, which is the rate limiting enzyme or the most crucial step in the fatty acid synthesis itself. Later, we will see another enzyme which is complex and has something like 8 components, and that involve in taking the product of this ACC, that is malonyl CoA, and making into fatty acid by using the 8 component. So here a little bit on the mechanism of acetyl-CoA carboxylase. This enzyme will take the acetyl coenzyme A and produce malonyl CoA in 2 steps. The first step is to take the carbonic ion and activate it with the presence of ATP, and once it's activated, the carboxy part of this carbonic acid or ion will be attached to the biotin to become carboxybiotin. This takes place in one active site of the ACC. Then the carboxybiotin with the long arm will be stretched and given to the next active site, which has the acetyl coenzyme A reacting with the carboxy portion, and then producing malonyl CoA as the product. So the biotin is the prosthetic group, the non-protein of this ACC enzyme, and very much relevant in the active site. Given here the full reaction itself, that is carbonic ion with the activation with ATP reacting to acetyl coenzyme A, and in the end producing malonyl CoA. And this happens in two reaction within two active site of ACC enzyme itself. Here shown the long flexible arm that allows the carboxybiotin formed in the first active site to be transferred to the second active site of ACC for reaction to take place with acetyl coenzyme A. And as you can see, the long arm, which is non-protein of the biotin, is attached to the protein portion by using lysine residue. Next, we look at the regulation of ACC. As referred to you many times, regulation mechanism of the enzyme is not going to be assessed in the final exam, and but basically you just know it for knowledge wise is excellent for me. In terms of the regulation of ACC, it is the committed step, and therefore the step that can be regulated, and it has an influence on the whole fatty acid synthesis as a whole. And how is it regulated? It is regulated by both covalent modification as well as allosteric control by local metabolites. In terms of the physical form of this activation, if it's in an active form, the conformational change takes place that it will look like a multimeric filamentous complex, meaning that few of these ACC comes together and become multimeric filament, and therefore that is the active conformation for this particular enzyme. And when it is inactive, deactivated, it will be in protomer or monomeric, meaning single unit. So here we can look into describing the whole process of regulation. The regulation takes place in terms of phosphorylation, adding and removal of phosphate. This enzyme, once it is removed from the phosphate, it will become active, while the adding the phosphate will make the enzyme to be inactive. And when it's inactive, it will be monomer. When it is active, it will be multimeric, has been shown in the diagram there in green. So that's covalent modification. So when is it activated and inactivated by phosphorylation and dephosphorylation? By the effect of insulin, when there is a lot of glucose, for instance, insulin will be secreted, and therefore with the lot of acetyl coenzyme A, insulin will cause activation of the protein phosphatase. This enzyme is capable of removing phosphate, and it removes the phosphate from ACC, therefore it makes the ACC become active. And that's how the acetyl coenzyme A, excess of acetyl coenzyme A due to excess of glucose can be then eventually converted to fatty acid itself. So that's activation. And then when there is low levels of glucose, meaning low levels of fuel, energy, therefore what has to be done is the reverse. And this is cause the phosphorylation of the acetyl coenzyme A. And that's done by AMP, activated kinase, and that will cause the inactivation of ACC. Normally, inactivation takes place when there is cellular stress. For instance, when the person is having a particular, say, flu, and therefore requiring energy to ward off the infection, or in terms of when a person is exercising, therefore they can break down the fats from the fat cells, and this requiring inactivation of fatty acid synthesis and activation of beta oxidation. So that's covering in terms of covalent modification by phosphorylation and dephosphorylation. Next, within this slide itself, we look into how the allosteric regulation or control takes place, where in the presence of citrate, it will cause the activation. Citrate is a product or intermediate in the citric acid cycle. When citrate is high, it's an indicator acetyl coenzyme A is also high in the cell. Therefore, to allow the acetyl coenzyme not to come into citric acid cycle, citrate will go and activate this ACC, therefore channeling or taking all the acetyl coenzyme to become fatty acid instead. And the palmityl CoA, on the other hand, is the product of the fatty acid synthesis. So this is a form of feedback inhibition, where the product, once it become a lot, it will be binding allosterically to ACC and stops its own production. So that covers in terms of how ACC is regulated, both in covalent modification as well as allosterically. So here you have the regulation described further in terms of ACC regulation. I'm not going to go in detail about the text as well as the diagram, because just only finished describing the whole process. But if you look into detail, you read the text, you will see that this slide, as well as this slide, is in fact describing this regulation, which in fact already been covered to you in the last lecture of fatty acid oxidation. But to briefly describe again in a different way, I just want to highlight when there is low blood glucose, meaning there is low energy molecule, therefore fatty acid will be tapped into in giving the fuel molecule. Therefore, beta oxidation will be encouraged. So how does that happen? It is by the secretion of glucagon hormone from pancreas, which cause the activation of protein kinase A. This protein kinase acts by adding phosphate. It will add phosphate to acetyl coenzyme A and therefore deactivating the enzyme itself, the rate limiting enzyme for fatty acid synthesis. When ACC is inactivated, it will cause less and less acetyl coenzyme A to become malonyl CoA, and therefore fatty acid synthesis is in a way inactivated. When there is low levels of malonyl CoA, it cannot go and inhibit the carnitine shuttle, and this allows the fatty acid from the cytoplasma to now go into the mitochondria and proceed into the beta oxidation. Therefore, energy can be attained from fat cells rather than glucose. On the condition of, say, there is high levels of blood glucose, what happens in the reverse? Insulin will be then secreted, and insulin is a hormone that will activate phosphatase, and this phosphatase will remove the phosphate from ACC and causing the ACC now to be active. When ACC is active, it will now cause the conversion of excess of acetyl coenzyme A due to a lot of glucose molecule to be malonyl CoA, and therefore malonyl CoA concentration increases, and this increase in concentration of malonyl CoA will cause inhibition of the transportation of fatty acid from cytoplasma into the mitochondria. Therefore, insufficient fatty acid will cause beta oxidation to be inhibited or not happening. And also, increase in malonyl CoA concentration will eventually be converted to fatty acid, and fatty acid will then be converted to triacylglyceride to be stored in the adipocyte as well as liver because of excess of glucose present in the blood. So that's another take in terms of regulation of fatty acid, where one is activated while reciprocally the other one is deactivated. This few slides to come actually repeats what I already described in detail. This slide basically talks about the insulin effect, where the increase in insulin causes dephosphorylation and therefore activating protein phosphate, and phosphatase will remove the phosphate for the ACC to become now active. So insulin causes activation of ACC, therefore fatty acid can be synthesized. Well, this slide describes of the feedback inhibition, where increase of the product of fatty acid synthesis, that is palmitoyl-CoA, much of its increased concentration will cause palmitoyl-CoA to bind to the allosteric site of ACC and therefore deactivating it, and hence no fatty acid further synthesis can take place. This in turn describes in terms of the high levels of citrate, which is the intermediate of Krebs cycle. Its high level of citrate concentration because high level of acetyl coenzyme A. So since acetyl coenzyme A is too many to actually go into Krebs cycle, since it's overburdening the Krebs cycle, what happens is the citrate will then activate ACC, and ACC will take up the acetyl coenzyme A to become fatty acid for storage instead. Not only that, cholesterol synthesis, ketone body production can be also encouraged, as well as making glucose through gluconeogenesis. So therefore, it reduces the burden of the acetyl coenzyme A entering the Krebs cycle. So next we're going to look into the fatty acid synthase reactions, and that takes place also in the cytoplasma. The the malonyl CoA that has been synthesized from ACC will now be utilized by fatty acid synthase in making palmitic acid. To talk about the fatty acid synthase, we talk about in terms of its presence in the prokaryote such as bacteria, and also in terms of its presence in the eukaryote mammal cells. In the bacteria, it has 7 components, that is 6 different enzymes and 1 acyl carrier protein. While in the mammal, the total components are together 8. In the prokaryote bacteria, it is actually uh, being in individual, not clustered. While in the mammal, it has been fused together due to evolution to be complex. So that's the difference. And in terms of fatty acid synthase, it requires the presence of NADPH for its reaction to take place. And this NADPH, the reductant, is supplied by pentose phosphate pathway. Here shown the fatty acid synthase in detail in diagrams. The diagram on the left shows that it is a homodimer with identical subunits, and each subunit has 8 components. If you do the counting, you should be able to get 8 components in each subunit. On the left, also should able to get 8 subunits, and two components which are very important in the growing fatty acid synthesis is the ACP and condensing enzyme. These two will be repeatedly used in growing the fatty acid, and then other components comes in modifying the fatty acid until it become palmitate CoA. This slide is just to show the difference between acyl carrier protein, one of the components in the 8 components for fatty acid synthase, whereby it has similarities to that of acetyl coenzyme A, where apart from having the thiol group at the far end on the on the left, it also has the long flexible arm, just like acetyl coenzyme A. So you have on the left the cysteamine, which has the sulfhydryl group, which can bind or cause formation of covalent bond and then get attached to anything that comes to it. You also have the phosphopentadiene, which is actually made from vitamin B, and then you have the big, huge part of the acyl carrier protein that is up to 10 kilodalton. So that's big. And then to compare with the coenzyme A, acetyl coenzyme A, you have also the long flexible arm, the sulfurhydryl or thiol group on the far left, and also on the far right you have the coenzyme which happen to have the negative charge phosphate, and this is the deterrent where the negative charge phosphate cannot bypass the lipid bilayer. As such, it cannot travel from mitochondria to cytoplasma that easily. So among the 8 component, the one that is mostly been utilized is these two, that is ACP as well as condensing enzyme. The condensing enzyme is also referred as the ketoacyl synthase. And these two, during the fatty acid biosynthesis, they help in the growing fatty acid synthesis, and basically they alternate between these two components before the addition of 2 carbon every time. So for this slide, I know it is looking very complex and create some anxiety for you to understand it. Let me try my best. Also to help you to see this whole slide, I'm going to use a highlighter for describing. But no worries, this overview of using a transparency type of slide is only the start off. There will be in the next few slides to come another animation to describe again in terms of the action of fatty acid synthase. So to begin with, it is the acetyl coenzyme A that comes into connecting with the fatty acid synthase. It comes in first time and only one time. After that, more of usage of malonyl CoA for the repeated step in the growing of fatty acid synthesis. So the acetyl coenzyme A will be connected to the ACP site as shown here. Next, the ACP will transfer the acetyl group towards the condensing enzyme as shown here. And after this, since the ACP is now free, it will then grab the malonyl CoA towards it as shown here, and then therefore now both the condensing enzyme and ACP has a group. These two groups, the acetyl as well as the malonyl CoA, will react and form the 3-ketoacyl group as shown here. But before it can react, the malonyl CoA will lose a carbon by forming carbon dioxide. This 3-ketoacyl ACP does not have the shape of the fatty acid yet because of the ketone group. So in order to modify the ketone group, three further steps will take place, and that requires the redundant NADPH, that is a reduction will take place here, okay, and then followed by dehydration causing the formation of double bond, and again another NADPH will be causing the reduction to finally form what looks like a fatty acid, that is acyl ACP. So now from 2 carbon it has formed to 4 carbon, but it doesn't stop there. The acyl ACP will, the acyl group will be transferred to the condensing site, and this will then carry on to form from 4 carbon to become 6 carbon through repeated process of this whole cycle for many more times until it forms the 16 carbon palmitoyl CoA. Here given a few slides to come in terms of describing in text rather than what I've just described in the previous slide, that complex explanation. So you can read about it. It will say the same as what I have described in length just now. This slide describe to you that the beta oxidation is actually the reverse in terms of the three-step, that is converting the ketoacyl to become acyl for the fatty acid synthesis, whereby in the beta oxidation it is oxidation, hydration followed by oxidation, if you still recall. But for the fatty acid synthesis, it is reduction, dehydration, and reduction. Of course, the reduction is assisted by NADPH, and that's obtained from pentose phosphate pathway. So here shown that repeated cycle will cause the extension from 2 carbon to 4 and then up to 16. The final 16 will be actually covered by the thioesterase, the 8th component of the fatty acid synthase, where it suddenly become alive and does its function, where in the product release. Thioesterase will form the final step, where it will release the palmitic acid from the fatty acid synthase. On that will be done as shown here, where assuming the 14 carbon is now formed at the ACP, and then to form the 16 carbon, it will then transfer to the condensing enzyme, and then the palmitoyl CoA will come, just like the way it's done for the extension, and the 14 carbon will be attached to the ACP. But what happens next is there will be no further reattachment of the 16 carbon to condensing enzyme, but instead it will come out catalyzed by thioesterase to form the palmitic acid. So that causes the product release of the palmitic acid by fatty acid synthase. So here the whole process is repeated again with a different animation slide, so that you can understand better compared to the other one, which uses the old transparency. So we start the explanation by having the fatty acid synthase component ACP, where it is free to react with acetyl coenzyme A to become acetyl ACP. But however, the acetyl ACP will transfer the acetyl group to condensing enzyme. So now ACP is free again, and that will be attached to malonyl CoA to become, as shown here, both the malonyl component as well as the acetyl will react and therefore become longer. The ACP now will have the ketoacyl group, and 1 carbon of the malonyl will be released out as carbon dioxide. The 3-ketoacyl group does not have the shape that is necessary for acyl or fatty acid. Therefore, further modification needed to be done. This will be done by firstly a reduction using NADPH as shown there, and then a dehydration takes place to form a double bond, and this lastly by another reduction with NADPH will create the acyl group that has the shape and structure of a fatty acid, but it's only 4 carbon. Therefore this process needs to repeat again, where the acyl group will be moved from ACP to the condensing enzyme. Okay. And now, without any further use of acetyl-CoA, but instead the malonyl-CoA, the malonyl-CoA will react with the growing fatty acid, and this whole process will be repeated until 16 carbon palmitoyl-CoA is being achieved. So here is the summary of what we have achieved. If you look into just one acetyl-CoA and 7 malonyl-CoA, that is the reaction done by fatty acid synthase, meaning to form palmitate using 14 NADPH. So you get the reaction as shown above. But if you take into account the ACC and also the reaction of fatty acid synthase, therefore you need to realize 8 acetyl-CoA will react with 14 NADPH, and 7 acetyl-CoA will be converted to become malonyl-CoA by using 7 ATP, and due to the 2 enzyme, finally you will get palmitate as the end product, the 16 carbon palmitate. Something which has been highlighted at the bottom is the issue where the acetyl coenzyme A is mostly found in the mitochondria. However, for the fatty acid synthesis, which so happen in the cytoplasma, so how does the product which is found in acetyl coenzyme A in the mitochondria need to be brought out from mitochondria to the cytoplasma? Because acetyl coenzyme A, having the negative charge of phosphate, cannot easily penetrate the mitochondria membrane, the lipid bilayer. For that, we need to look into the next slide, where citrate shuttle will be utilized in taking the acetyl coenzyme A from mitochondria into the cytoplasma, where it can be then utilized in the fatty acid synthesis. So here the citrate shuttle is described in detail, but not for you to remember as it is not covered in the final exam. So in terms of how acetyl coenzyme A is brought from mitochondria into the cytoplasma is firstly it needs to be converted to become citrate. Citrate can then escape from mitochondria into the cytoplasma, and then once this is in the cytoplasma, it can be reconverted back to acetyl coenzyme A. So now the acetyl coenzyme A is now found in the cytoplasma. The breakdown of citrate will produce oxaloacetate. This oxaloacetate cannot penetrate back into the mitochondria, therefore it needs to be changed into malate. Firstly, malate can go back to the mitochondria, but malate also can be converted to pyruvate. So both pyruvate and malate can therefore be brought back into mitochondria, and they can be converted to oxaloacetate. This oxaloacetate will react with acetyl coenzyme A and form citrate, and this process of citrate shuttle will carry on to bring more and more acetyl coenzyme A from mitochondria to cytoplasma for fatty acid synthesis. More on the regulation, and this time regulation on the fatty acid synthase. The ACC, we already look into its regulation, and that is, since it's the rate limiting step, it's been regulated by covalent modification as well as allosterically, which is normal of any rate limiting enzyme. But for this fatty acid synthase, which is not the rate limiting enzyme, it is actually regulated by producing it. That is making fatty acid synthase by transcription and then followed by translation. So it's a bit slow, where it needs to be generated itself by using its gene. So for that, we look at firstly the liver and later in the adipocyte, how it is transcribed, how the insulin can simulate the fatty acid synthase expression itself. And that is done by activating two factors, that is firstly upstream stimulatory factors, and another one is the protein itself, the SREBPS, that is sterol response element binding proteins. These two components will, at the end result, will be causing the fatty acid synthase expression to be activated, and therefore fatty acid synthase will be made. And a bit of information given at the bottom text there, where it is been found that fatty acid, polyunsaturated fatty acid, those Eskimos taking fish oil, as well as the Mediterranean people who take olive oil, has a lot of this polyunsaturated fatty acid. It's been studied that it can inhibit the SREBPS activation. If it can inhibit, therefore the sterol, that is cholesterol synthesis, is also inhibited. So this sort of like gives some evidence that this population having low consequences of heart problem, because cholesterol is low in their body due to their diet itself. So in the fat cell or adipocyte, there's also regulation on the fatty acid synthase. This time it is by the hormone leptin. Once the hormone is secreted, it causes the uh, response as shown here, where it regulates the decreasing food intake. It causes the person to be less likely to be hungry. It also causes more active of the person to lose energy, so it increases energy expenditure. Leptin also causes inhibition of fatty acid synthesis. So at the end, the person will less likely to become obese because of these effects done by leptin. Interestingly, this leptin has been studied for the purpose of losing weight by creating it as a supplement for people who need to control their weight. Next, we look into elongation. That means it's from 16 carbon, how to make into 18 carbon, and so on. And then later we look into desaturation. That means adding double bond to the fatty acid that is being formed. Here the elongation is being described in text where it states that the elongation takes place in two locations, that is the mitochondria as well as the smooth endoplasmic reticulum. To describe the process of elongation is best to see from this next slide, where the elongation takes place in two systems, that is mitochondria and also the smooth endoplasmic reticulum. So assuming now you have the palmityl-CoA 16 carbon already been formed by the effect of ACC as well as fatty acid synthase, it now moves into either mitochondria or smooth endoplasmic reticulum from cytoplasma. Then comes the malonyl-CoA in the smooth endoplasmic reticulum. The palmityl-CoA will firstly lose its coenzyme A, while the malonyl-CoA will become less by one carbon to be carbon dioxide. Both will react and form the 18 carbon sterile CoA, right? That's in the smooth endoplasmic reticulum. While in the mitochondria, the acetyl-CoA comes in instead of malonyl-CoA, and again after the palmityl-CoA loses its coenzyme A, it will react with the acetyl-CoA to form the 18 carbon sterile CoA. What happens next in the smooth endoplasmic reticulum as well as in the mitochondria is slightly different. In the smooth endoplasmic reticulum, the reduction is done by NADPH, while in the mitochondria reduction is done by NADH. But the further steps are similar both in the mitochondria as well as smooth endoplasmic reticulum. Therefore, you will now have the sterile CoA by both of these systems. So after the elongation, that is forming of sterile CoA 18 carbon, certainly need to add a double bond to become oleic acid. So to do that, it needs to have certain essentials such as the desaturase as well as carrier protein. This desaturation takes place in the membrane of the smooth endoplasmic reticulum. So once the elongation takes place in the smooth endoplasmic reticulum within it, then it moves to the membrane to cause the double bond to be formed. The certain issue is the saturation beyond uh, position 9, that is 12 and beyond, cannot be done in the mammal because there's no desaturase for 12 and beyond. It only has the desaturase enzyme up to position 9. Therefore, the mammalian cell need to obtain any fatty acid beyond double bond at position 9 and beyond as essential fatty acid. It needs to be only obtained from diet, while plants can make all the fatty acid, even those beyond the position of 9. So here shown the requirements for desaturation of fatty acid. You need to have firstly desaturase, particular desaturase, whichever position to be present, and also you need the carrier electron carriers, that is two been shown here, NADH cytochrome B5 reductase as well as cytochrome B5. These are the carriers of the electron and also shown the desaturase. Here shown the desaturation, what needs to be done. Basically, you need to have the smooth endoplasmic membrane, endoplasmic reticulum membrane, where the desaturation takes place. And then mammals unfortunately only have desaturase for position 4, 5, 6 and only 9. Beyond that it's not found. Therefore it has to be called essential fatty acid and obtained from diet instead. So here shown the process of desaturation at the smooth endoplasmic reticulum membrane, where it has desaturase. Firstly, the NODH will be releasing its electron to the carriers, and until it arrives at the oxygen. Unfortunately, oxygen cannot be reduced to become water by the effect of NADH alone. To enable it to be reduced, it also needs the help of saturated fatty acid. Once the saturated fatty acid releases its electron and also accompanied by hydrogen, the oxygen can now be converted to water, and in the process saturated fatty acid will be incorporated with a double bond. As such, it will become unsaturated fatty acid. This is the another representation of that desaturation process, where shown that the membrane of smooth endoplasmic reticulum has the two carriers of electron as well as the desaturase within it. So it starts with the NADH providing the electron as well as the hydrogen ion going through the carrier proteins one at a time, and then finally to the oxygen. I need to apologize that this animation is missing one more of the what do you call the arrows, which supposedly should come from here to here and indicate that the oxygen is being reduced to water, and also the fatty acid 18 stearoyl CoA will now become 18 carbon allyl CoA, that is unsaturated fatty acid. So we come to the last slide on the lecture of fatty acid synthesis. This slide sort of like summarize what happened, what we have studied so far, where we start off with forming the palmitic acid, which has 16 carbon, with the aid of ACC first, where it forms lots of malonyl CoA from acetyl coenzyme A, and in the use of fatty acid synthase, this malonyl CoA as well as one acetyl CoA can be utilized in forming at the end palmitoyl CoA, and that will be then utilized in the making of say stearate or stearic acid by elongation. And this is while palmitate CoA is done in the cytoplasma, the stearate or stearic acid is done in either mitochondria or smooth endoplasmic reticulum. So two systems, but the end result is forming or elongating the palmitic acid. Apart from elongation, also possible to add a double bond by using desaturase, and that's done in the smooth endoplasmic reticulum membrane with the help of desaturase for this desaturase location 9 is required, and that's found in mammal, no problem. And even the formation of oleate is also possible to be done by mammal cell because there is the desaturase 9. However, beyond this line, all those fatty acid cannot be formed by mammal cells because they do not have the desaturation relevant to form to form double bond at position 12 or position 15 and so on. All these fatty acid can, however, be formed by plant cells. So mammals need to rely on diet of plant such as vegetables to actually obtain essential fatty acids.